Assalamu alaikum. Welcome to this live stream. This is part 5 of our series on orphan genes. I'm so grateful to have Paul Nelson back on the channel to be giving us some really novel information and insight into the world of orphan genes. So welcome back, Paul, to the channel.
Thanks for having me back. I'm looking forward to it.
Brilliant. So over to you. We have some a very interesting title here, and it's going to be a novel session, hopefully.
Yes, this is material that I haven't presented on this channel before, but it is relevant to putting the context in place for an intelligent design understanding of orphan and taxonomically restricted genes, which is what I'm working towards. That will be the conclusion of this series. But to use the metaphor of a puzzle, this is a piece that needs to go into place before we finish the puzzle. It's necessary to give the full context of what I want to say at the end. All right, so the question we'll be addressing for the next hour or so is how to investigate biology from an intelligent design perspective when mainstream opinion, that's what I mean by everyone here, it's obviously not everyone, because I disagree with this. When everyone says doing that is obviously impossible. So let me refer to a friend of mine who is a microbiologist at the University of Idaho, Scott Minnick. He works on the bacterium that causes the plague, Yersinia. And I met him almost 30 years ago when I was still a graduate student, and he told me something that I've never forgotten. He said, Paul, none of this intelligent design debate should be that controversial. And he went on, the reality, whether anyone is conscious of it or not, is this. Most molecular biologists are de facto design theorists already. In other words, the actual practice of molecular biology, genetics, and other sciences of biology presuppose an intelligent design framework, not by name, obviously, or this idea would not be so controversial, but in its content, the central notions that people were employing in their reasoning really only made sense within the framework of intelligent design. But you might ask, well, how could that be possible? Design was and is widely viewed as intrinsically unscientific because it employs a cause, a transcendent designing intelligence that is unobservable in principle. So Scott wasn't daunted by this. He said, look, the actual practice of molecular biology presupposes concepts that really come to us from design. So I want to lay out what he meant in this presentation and unpack that for you, unpack this intuition that Scott had. And I hope by the time we reach the end, it'll make sense to you. So quick outline. I first want to discuss what I call the basic logic of functional triangulation, which is something that you've done. You may not give it a name, but the inference pattern that I'll be describing is something that I think everybody uses at one point or another, even in just sort of their daily affairs. Then we'll focus in on functional triangulation in biology. We'll look in some detail at what I'm calling the adapter hypothesis, or it's not my name. That idea was due to Francis Crick and his office mate at Cambridge at the time in the late 1950s, Sidney Brenner. So part 3, Crick, Watson, and the adapter hypothesis. Well, we'll look at functional triangulation in a sense that's or in a setting that is directly relevant to intelligent design. And lastly, we'll answer the question, why is functional triangulation properly called design triangulation? And look at a notion that I'm going to call the causal primacy of the organism. And the goal throughout all of this, as I've said, is to show how design illuminates biology. In particular, how design conveys an explanatory advantage that you don't get from any bottom-up perspective. Design uniquely of all hypotheses on the table in 2023 allows a top-down system-first perspective that I think from a naturalistic or materialistic framework is impossible. So I hope to prove that, or at least to make it plausible to you as we go on. All right. Basic logic of functional triangulation. So this actually happened to me. An embarrassing episode several years ago involving my daughters and their badminton team. They both played badminton, high school varsity sport here in Illinois. So let me give you the context. Oh, here's one of my daughters. This is Olivia. She was a very good badminton player, and she and her team were going to a tournament in downstate Illinois. So Chicago, as you know, sits at the northeast corner of the state of Illinois, and I had to take them, a few hours downstate to a tournament. So here's my car at the time. It's a Honda Odyssey minivan. And I'm here, and the badminton team is sitting in the middle and back rows. And we pull into a gas station for two reasons. One, I needed to get gas in the Odyssey, but they needed to use the bathroom, the badminton team. All right, so I pull in, I grab the button that opens the gas tank. And then I can't open the left door, this automatic sliding door right here. It won't open. And the team is saying, Mr. Nelson, please let us out. We need to use the restroom. All right, so I can't get the door open. I'm utterly mystified. So I reach over and I grab the owner's manual, which tells me this. Before refueling, make sure the rear sliding door on the driver's side is closed. Why? Why Honda Corporation? Well, here's a clue. This is why. Okay. The Honda Corporation has built the car in such a way that I will not be able to do something that's physically impossible, namely have two macroscopic objects with mass, like doors, occupy the same location at the same time. So you can see the fuel door is here. This opens on a little hinge, and with a tube line going down to the gas tank. So that's where I would put the gas in. And here's the door. And you'll notice that if I try to open this door while this is open, I'm going to have a problem. In fact, the problem, I guess you could say, would be sufficiently severe that the Honda designers have planned for it. Okay, so this is actually taken right out of the owner's manual. When you release the fuel fill door, the driver's side sliding door automatically locks. All right, that's why I couldn't get it open. Why? Because it may crash into the fuel door. And the designers say, there are going to be some people who buy this car who are a little dim. And they are going to try to open that left sliding door when the fuel door is also open. What do we do as engineers at Honda? We build a system so that when you hit this button, as I did in my position in the driver's seat, there's a logic circuit that says, wait a minute, is this open? If it is, we're not going to let you open the fuel door. We're going to stop that signal from going to the we're not going to let you open the sliding door. We're going to stop this signal from getting here to prevent, a catastrophe of the door jamming. The two doors would jam on top of each other. All right, so that's the background. So I have a younger brother, Peter, who's a doctor, very, very bright, very competent. I do not consider myself a competent person. In many respects, I'm not at all a competent person. But Peter is sort of competent across the board. So he's looking at my car. He doesn't own an Odyssey, by the way. He drives another vehicle. He's standing in the driveway of my house looking at this car, and he notices this, that there's a potential conflict here. And he makes an inference. He triangulates, all right, like this. The left sliding door moves, or excuse me, the sliding door moves to the left rear over the fuel door. That's fact A. Fact B, if the fuel door were open, the sliding door would jam. All right, so A and B are two facts that Peter has established just from observation about this vehicle. And he says, it's probably the case that the Honda engineers have taken account of this. They know A, they know B, they know that some Odyssey owners like Paul Nelson are a little dim, not always thinking properly. That, you know, we need to help them to keep them from damaging their own vehicle. So there's probably a locking mechanism to stop the sliding door from jamming. Now, strictly speaking, this is not an existence proof, but it does confer a high probability on the existence of C. All right, if you think about the complex systems that you use, like your laptop, there are many functions in, let's say, the word processing software in your laptop, such as the ability to reverse a decision you've made, where you delete something and you think, oh, I didn't want to delete that. Well, I know in Word, the word processor that I use from Microsoft, I can go up and there's across the bar on the top, there's an option in a pull down menu to reverse that decision and restore what I deleted. In other words, I cancel the delete and the text comes back. There are many features in human design systems that account for contingencies, errors, accidents, and so forth. You have probably on your laptop or on your computer automatic backup. Why? Because you might lose a file, or you can't find a file, and what you need to do is go into your backup archive and retrieve it. In other words, systems are designed to account for these kinds of contingencies. It's built into the system. All right. We can discover these features simply by triangulating, in the sense that we know facts about a complex system and we say, given A, given B, it's probably the case that something like C exists. Well, we can use this as a metaphor for scientific inquiry, right? We know A, we know B, something like C is probably the case. So we do this in daily life, and we do it in science. And it's a research strategy. You can think of complex systems and their features like black boxes, where the black box metaphor, as you know, conveys the sense that there's a lot of information within that black box that one cannot see directly. But we can infer its existence by doing what I've just explained, by triangulating from what we do know to what we haven't yet seen. So here we have A, fact B. These have an entailment relation to each other. These are connected facts. They're not stray observations. There's some important connection between them. And we infer a part system or functional relation that must exist even if not yet observed. All right, given the strength of our inference, in other words, we think, well, this is a pretty strong triangulation. We're pretty sure C is there. That provides a warrant, a research warrant, if you will, to go looking for C. So you can think of this functional triangulation as a research strategy that motivates investigation. And as I've said on other streams with Subor, science is hard. Nature hides her secrets, right? If science were easy, we wouldn't need labs. We wouldn't need instruments. We wouldn't need peer review. We wouldn't need the things that help us towards the truth and that enable us to find it. So science is hard. So you want heuristics that are reliable, that tell you, you know what, you're not going to waste your time if you go looking for C, because you're sure of A and you're sure of B, and you know that these facts have a relation to each other. very probably C is there, and that warrants or underwrites the search, the research, the going to look for it. All right, so that's the basic notion of functional triangulation. Now let's apply it to biology. All right, let's get our 2 facts on the table. Copper is a poison. You do not want otherwise unemployed copper ions sloshing around the cytoplasm of your cells. It's a highly reactive metal. It can create all kinds of mischief. If you have too much copper in your diet, or you somehow are exposed to too much copper, lots of bad things follow from that. So that's your fact A. Copper is a poison. Fact B, copper is an essential cofactor in many enzymes. You have to have it. You have to have a certain amount of copper in your diet. If you don't, these enzymes aren't going to function properly, because that metal is essential for the role of these enzymes. So here is cytochrome C oxidase, and you can see that there are copper atoms here. There's a copper atom here. That metal is essential for the function of this enzyme. All right, who can do this? Trust the logic, triangulate. So we've got our fact A, free copper is highly toxic to cells, but you have to have it. So A and B both refer to copper. They both refer to cell function, organismal function. In one case, too much copper, otherwise unemployed, as I've put it, is a poison. B, you've got to have it. Now, from those 2 facts, you can triangulate. What must exist in aerobically respiring organisms like you or me? All right. I've done this with classes, and usually there's a few moments of mystification. They're like, well, I don't know what Paul's getting at, but I can't follow this. So I do it very literally. I say, think of my fist as a copper atom, and over here, let's imagine a cell membrane. And I've got to get this copper to where it needs to be. What kind of a system do I need? And if I make it very literal like that, they say, well, you're going to have to grab that copper, right? I've got to bind it. What else do I have to do? I've got to transport it across the membrane into the cell, and then I've got to transport it to where it needs to be, and then release it in a controlled way. So they can see from A and B, from poison and essential, there must be a system that does this, that, as it were, is the chaperone or the control system for the transport of highly reactive metal ions. Now I have a student in the class, or maybe one of you guys watching this right now, and you say, well, how can you be sure it's there? This is how. Okay? Cells are not working by magic. They're not. They are using systems of incredible sophistication to do the tasks that they need for function, for viability. Don't worry, it's there. So we can call this the no magic principle. Organisms make use of mechanisms for their very existence. Whether we see those mechanisms or not, they're operating. Okay, so the no magic principle is just another way of phrasing this notion of functional triangulation. When I couldn't open the left sliding door on my Honda Odyssey, it wasn't magic that was keeping that door locked. It was a designed feature of the car as a whole, that said, we're not going to let you jam the door, Paul. If the fuel door is open, you will not be able to get that sliding door back. And you could say, I could go looking now in the circuitry of the Honda, in particular, the electrical circuits that control the doors, for that feature. I know it's there. Okay. Now, what's interesting about this, what I'm going to call here an existence proof for a copper binding and transport system, what's interesting is this was known long before the system was actually observed. So in this article in Science from 1997, the authors say, what do we know about copper? Well, what did we know? What we did know was you needed it for aerobic life because of its role as a metal ion cofactor in many important enzymes. What else do we know? It's a poison. How do you rationalize those two, A and B? You assume that there's a system that cells are using, that organisms are using to control their copper. Okay. We don't know where it is. We haven't characterized it. We have a pretty good conviction, a pretty good inference that it's there. Okay. Same article, they go on. The agents, that is, the actual molecular players of such trafficking, have remained largely unknown. So you can see how this mode of reasoning warrants or underwrites research. You say, well, something is doing that job. It's got to be in there. Let's go looking for it. We have a strong sense it's got to be there. And here it is. So the article I was just quoting was a news story in Science about this discovery. And this cartoon shows the features that cell biologists knew had to be there. So here are these red dots are the copper ions. Here's a portal through the cell membrane. And I won't go into all the details. I'm going to send the references, of course, to Subor. You can look them up if you want. But once the copper gets within the cytoplasm, it's not free to create mischief. Highly reactive metal is bound immediately and transported to where it's needed and then released. And this portion of the cartoon shows the mechanism of these proteins in action as they're sort of handling this highly reactive metal as they pass it off into its enzyme location where it needs to be. Now, when you read the article describing this discovery, there's some very striking language in the article itself in Science. And I'll just quote it here. These chaperones, this copper binding system, guides metal ions to their appropriate biological partners and protects and keeps them from being trapped at adventitious sites. That means places where the copper can do mischief. Our results underscore the idea that cells make use of elaborate machinery for recruiting, trafficking, compartmentalizing, and inserting into the appropriate enzyme reactive cofactors such as mononuclear copper ions. I love this next phrase. This is unlikely to proceed by spontaneous self-assembly. Rather, metal insertion is emerging as an orchestrated event. So this is my first of several examples of biological triangulation where you've got two, or obviously n, you could have many facts about some black box. And from those, you draw an inference to a system that must be there, even though you haven't yet observed it. So the history of biology shows that this method works. Now, here we need to shift out of biologist mode and go a little bit into philosopher mode. In other words, step back and let yourself reflect on the logical structure of what's going on here. I guess you could put it this way. How do you know that this works? Or what must you presuppose to get this mode of reasoning up and running? What's necessary to get this started? I'll answer that question in a few minutes, but begin to think about that. What do I have to presuppose to make this kind of top-down inference? All right, thought experiment. Non-biological thought experiment, but complex system. All right, so living things fall within the class of complex systems. So this will be a complex system inference. Suppose a friend gives you a black box that does the following. 8 a.m. every morning, it plays the music of J.S. Bach, but only on even-numbered days. Okay, so it wouldn't play, today's the 25th, is that right? Yeah, today's the 25th of July. It wouldn't have played Bach today. Yesterday, right? Monday, you would have got, let's say, Bach's First Prelude. On odd numbered days like today, you get the Beatles, Hey Jude. Gosh, so many beautiful rock tunes from the Beatles. Who knows? Maybe Penny Lane, or pick your favorite Beatles tune. Only on odd numbered days. And it's invariably accurate. Whatever the numerical odd or even value of any particular day is, you get either Bach or the Beatles. And this is what the black box the black box does. So at this point, you're just observing this funky little toy that your friend gave you. You don't know how it works, but it obviously does, because you track it for, let's say you track it for a month, and it always gets either Bach or the Beatles, depending on the calendar. Now, you haven't opened the box. You haven't, unscrewed it, unscrewed the base and looked inside to see what the circuitry is and so forth. But just from your observation of what the box is doing, you can infer the existence of certain things, such as an accurate clock and perpetual calendar, because it's always hitting at 8 a.m. for your particular time zone. Okay, and it's got something that is tracking the calendar. Something is storing the music of Bach and the Beatles. Something is doing the scheduling, some bit of software, let's say. And of course, then you have some physical requirements, such as a power supply, amplifier, speaker, and so forth. Okay. Now, there are many possible ways that this could happen. We call this, in the philosophy of biology, multiple realizability. Functional analysis, what we've just been doing, that kind of top-down functional analysis, tells us that the parts must exist, but it leaves open the possibility that they could exist in a variety of ways. So you could have, for instance, a radio transmitter being sending you from some other location that music, and the box is just a receiver. We know that somewhere those entities must exist that correspond to these four functions that we've observed. Or excuse me, the functions tell us that the entities exist, but the entities could have multiple forms. Having made that caveat, we can go step back and say, what is the logical structure of the inference we're making? What's primary? It's not the parts, because we actually haven't seen them yet. So what we're doing logically is we're starting with the system as a whole, and we're inferring the existence of its parts, but we start at the top level and work our way down. We are not starting bottom up. Now, this kind of mode of reasoning is a strong challenge to a materialist or physicalist conception of causation, where, let's say, at the origin of life, it wasn't a whole cell that started life. It was the bits and pieces. In the RNA world scenario, it was a ribozyme that was able to make high fidelity copies of itself. In a metabolism first origin of life scenario, it was some primitive or early quasi-metabolic pathway, some chemical pathway, let's say in a submarine vent, and that somehow had a genetic system associated with it. In other words, all of these physicalist, materialist, bottom-up scenarios begin with the parts, not the system as a whole. This is very important. In fact, you could say it's the primary takeaway from this presentation, that intelligent design as a causal explanation enables you to start with the system as a whole. Why? Because that's what designers do, right? They conceive and visualize or represent conceptually, not physically, conceptually, a complex functional endpoint, and then they bring everything physical necessary for that endpoint into association to enable the endpoint to exist. So the direction of causality for an intelligent design theorist can begin with a whole system. All right, so keep that in mind, because it'll be very important as we come to the end. Now, you have to have the target state. You can't start with the parts because from the lower level, you can go in all kinds of directions. So here I've just shown a bunch of speakers, transistors, a little motherboard there and so forth. Ask yourself this question: what higher level system with its unique functions is entailed by the existence of these lower level parts? And you say to me, well, Paul, I can't answer that because I can go in any number of directions given these parts. Maybe I'd get a loudspeaker, right? Maybe I'd get a car radio. In other words, I can go in lots of directions from the lower level. I won't end up necessarily at the black box that you showed me in the thought experiment. There is a logical asymmetry here. Because of multiple realizability and the many-to-one relations that exist between parts and wholes, finding a higher level target state is an intractable problem if you don't have it already, right? If you try to start with the bits and go towards the target state, the complex system with unique functions, it's going to be very difficult because you can go in lots of directions, perhaps none of which is the target state that you want. The space of possibilities is too large. Okay, so logical takeaway from this: the highest level underwrites inferences to the existence of its parts, but one cannot go in the other direction. Without the functional whole as a target to guide us, we can't pick out this particular black box from all the possibilities that exist when we consider the parts that make it operate. This is clearly visible when you look at origin of life research. And I'll just do a little segue into origin of life research, and then we'll get back to design triangulation. So in 2020, a fascinating paper was published in Science on a computer simulation where the authors began with a starting set of plausible early Earth chemicals: nitrogen, methane, ammonia, water, hydrogen, and so forth. And then they ran the simulation to generate all the possible products from the interaction, the undirected interaction of those starting chemicals here. So here's our starting set. And they let the chemistry run itself, basically, without specifying a target. How many possible interactions are there among these starting points? So here along the vertical axis, you can see the number of generations where when chemicals combine, then you let them recombine and so forth. And they haven't shown the seventh layer because the seventh layer, so these lines going up are the interactions among these chemicals. They haven't shown the seventh layer because it's enormous. The number of products just explodes. But look at this ratio. 53 of the products from these starting chemicals were biotic. That is, they were biologically relevant. Over 34,000 were abiotic. Okay, that should make you think. That should really give you pause. Undirected chemistry does not head towards the living state. It's going to produce everything and anything. Let's look at the ratio here. 53 of the products were biologically relevant. Over 34,000 were not. What does that mean? What that means, among other things, is that living things are highly selective with respect to chemistry. For instance, amino acids. Chemical processes that produce amino acids produce hundreds of amino acids. There are 22 if you count selenocysteine and pyrrolysine. There are 20 amino acids, let's just stick with that number for the moment, in the proteins that we find in living things. What happened to all the other ones? Of both hands, right? Left-handed and right-handed. Something selected them out and keeps them out. Bottom-up chemistry does not know where it's going. It doesn't know that it's supposed to build a cell. It just does what it does. So the top-down perspective on the origin of life says, look, what we find, the chemicals, the biochemicals or biomolecules that we find in living things is a tiny fraction of the set that would be produced by undirected chemistry. And in the same paper, the alchemy simulation paper, in the supplementary materials, they say, some of these molecules like ATP, they're big and complicated. The problem is undirected chemistry produces very large numbers of molecules that you don't want. This is actually, this extremely here that's underlined on the slide, that is their emphasis in the paper itself. You get extremely large numbers of other not very interesting molecules if you're just looking for ATP. So a visual metaphor for this would be a heap of sand particles, sand grains, who knows, a million, some big number, right? One of those is ATP. Good luck finding it. All right. This metaphor of a few or one specific grain in a sand pile gives you a sense visually of what's happening here with chemistry and the need for selection, some kind of selective process right from the start. And this has long been known by people who work on the origin of life, who have no sympathy for intelligent design. Francis Crick, for instance, lifelong atheist. In fact, he went into what came to be known as molecular biology because he wanted to show definitively that there was nothing special about the living state that distinguished it from chemistry and physics. Living things were just souped up chemistry, complex enough, the right kind of complexity to make high fidelity copies of themselves. But analytically, when you get down to the nuts and bolts of what life is, it's just physics and chemistry. So in his book, Life Itself, in 1981, where he proposed the directed panspermia hypothesis, which he also published actually in the peer-reviewed literature with Leslie Orgel, he says, what is going to do the selection before we have the enzymes that in living things that we know are doing this for us? In this passage, for instance, he says, the precursors to the biomolecules that we want need to be separated. We need to select them out. And chemistry itself is not going to do that because chemistry itself is going to give us a whole lot of stuff, only a tiny fraction of which is biologically relevant. Robert Shapiro, another origin of life researcher who was, I guess, well known as a skeptic of many of the dominant hypotheses in the field, he says, when you have chemicals that are just doing what chemistry does, I keep using that phrase, but what I mean to convey is it's an undirected process. You get haphazard mixtures and chaotic polymers. In the Miller-Urey experiment, for instance, most of the yield of that experiment, the longer you let it run, most of the yield is insoluble tar that coats the inside of the reaction vessel. It's not, you're not getting a nice selection just of left-handed amino acids in the set of 20 that life uses. All right, so that was our little digression into the origin of life. But this kind of evidence points people towards the triangulation method, in the sense that if you start top down, if you start with the system as a whole, it will tell you things about itself that you would never get bottom up, where the system is not yet there. Another example, William Harvey, great naturalist of the early 17th century. And at the time that Harvey was working, the authority of Galen dominated medicine and biology. And Harvey's reading Galen, and he says, does Galen's account of the movement of the blood actually explain what we observe? Now, this is very hard for us in 2023 or post Harvey, let's say, to conceive. But in Galen's system, it was unidirectional. The heart, the lungs, the liver, they kind of watered the tissues, right? Galen knew the blood had to move, but it moved unidirectionally. And I won't try to go into how his system worked, because in fact it didn't work. And Harvey was able to establish that. But what was important was Harvey said this unidirectional flow, we can test that. Using sheep, using pigs, using domestic animals, we can test whether unidirectional flow actually works to keep an organism alive, to keep actually a complex vertebrate like a dog or a sheep alive. So a commentator writing on this said, what did Harvey do? What was the structure of his reasoning? Well, he used teleology. He started with the system as a whole, which is characteristic of teleological reasoning, and he reverse engineered the system, not as a final proof, but rather as a heuristic, a means to establish testable premises. So he triangulated beautifully. Started with a model of the function of the heart and said, let's suppose the left ventricle, so the blood's going out of the heart, discharges or pumps 3 ounces of blood per beat, and let's pick a rate of 33 beats a minute. The total volume of blood pumped with those parameters would be enormous. But he measured the total blood volume in dogs and sheep. They gave their lives. I mean, I'm very sentimental. I would find it hard to do this kind of research, but he bled dogs and bled sheep, bled them total of their total volume, of total blood volume, and measured that. And it didn't exceed a few pounds. So Harvey takes fact A, his estimate of how much blood would be pumped, 3 ounces, 33 beats per minute, during the life of one of these animals, and said, well, how much blood is there in that animal? I can open their arteries and bleed them till they're dead, sadly, but I can measure the total blood volume. It's way below what would be produced by that pump in A. And he triangulates. From A and B, he says, the blood must circulate. Galen's system, his ebb and flow irrigation, unidirectional irrigation of the tissues, it will not work. And what he said is just beautiful. He said, look, it's a matter of necessity that the blood performs a circuit, that it return whence it set out. No magic. What kind of necessity is he describing? Well, it's functional necessity, not physical necessity. It's a systems level property. It is a property of being a dog, that when the blood leaves the heart, it's going to make its way back. It's going to circulate. Because dogs and sheep and humans are not operating by magic. They're operating by principles that we can understand and characterize and test once we grasp them, if we take that no magic principle seriously. And the remarkable thing is living things do the math for us in a sense. Why? Because they are alive. And viability as the governing or top state in our hierarchy of relations tells us, if you say no magic, then it's got to work somehow, and we can triangulate then to the existence of systems that we haven't yet seen, such as blood circulation. The organisms, in other words, give us the clues that we need. Now, Harvey was resisted by his contemporaries for several interesting reasons. Probably the most interesting of those is if you think about a circuit, right? So you start at A, and let's say the left ventricle of the heart, and you're going to come back to the heart. That's a closed loop. That is something where you've got to have connections all the way around. In order for the blood to circulate as it makes its way out into the tissues, it's got to have a way to get back. But at the time, microscopy was in a very primitive state, and no one had yet observed capillaries. Capillaries are, you can think of them as on a metaphor with a highway system. I've got here in where I live in Chicago, I've got the federal interstate system, 8 lanes, 4 going north, 4 going south. I get off, I get on another smaller road that's 4 lanes. I come closer to my neighborhood, the number of lanes drops to 2. And then right outside my house, there are some one-way streets, right? But those connect back to the 2-lane. In other words, the circulatory system for the city of Chicago, Cook County, Illinois, has all kinds of routes that if you trace them, will allow you to go in a complete loop. It's not a dead end. Capillaries are the lowest level of that system in any animal with a circulatory system. They're the lowest or the smallest connection. They hadn't yet been observed. So Harvey's contemporaries said, well, how can the blood circulate when we don't see that connection? Well, they exist. Capillaries are real, but you need the right technology to find them. In other words, Harvey inferred something that had all kinds of downstream consequences for observation, which is what you want a science to do. You want a science not just to find fact A, or as I'm using with my triangulation metaphor, fact C, but fact C should entrain fact D and fact E and fact F and so forth. In other words, it's fruitful because it leads you to knowledge that you had no idea would be there. All right, another example. I've got to move more briskly. Oh gosh, too many digressions. I'm going to skip this example just for the sake of time. I'll provide the citation to Subor. I apologize for skipping it. It's a great example, but we really have to stay on track. All right, Crick, Watson, and the adapter hypothesis. Okay, this is where the temperature in the room begins to rise, and this is the case where or the example that is where we move from functional triangulation to why I want to call this design triangulation. Okay, our players in our story. Here is our protagonist, our hero, Francis Crick. Here's James Watson, who will be the antagonist or the villain, so to speak. He's brutally honest about himself, almost to a fault. So we're going to talk mostly about Crick, and then we'll get to Watson. All right, 1955. Forget all the cell biology you know, all the molecular biology. Put yourself back in the mid-50s. You're a biologist. What do you know? Let's say you're in a group that you're working with Crick on the mechanism of inheritance, genetics. Well, you know that DNA carries information. It is the primary information carrier in all living things. But look at the chemistry of DNA. You've got a sugar phosphate backbone as sort of the legs of the ladder, if we use the ladder metaphor. And then you've got hydrogen bonds across the double helix as the rungs of the ladder. But this chemistry is very regular. And actually, as chemistry, a little bit boring. When you need something to specify very different amino acids. Here are 8 of the set of 20/22, depending on how you count, present in proteins. Unlike DNA, the sugar phosphate backbone, hydrogen bonds, amino acids are very different. Some are polar, some are not. Some are big and bulky. Others, like glycine, are rather small. What is mediating information transfer from the very regular chemistry of DNA to the very irregular chemistry of amino acids? So this is the puzzle that, following the elucidation of the molecular structure of DNA by Watson and Crick in 1953, the work for which they eventually won the Nobel Prize, this is the puzzle that they're trying to solve. What is mediating information transfer from these two realms? The realm of nucleic acid to the realm of amino acids and proteins. So Crick was a member of what was called the RNA Tie Club, a very select group of biologists and an astronomer, George Gamow, eccentric, brilliant astronomer who was one of the spark plugs for this group. The RNA Tie Club, they got ties, actual physical ties. If you look in Google Images for RNA Tie Club, you'll get a picture of some of these guys wearing their very jaunty ties. And they were trying to solve the coding problem, the problem I just described of information transfer from nucleic acid to protein. And Crick contributed a manuscript. What you see, the cover sheet there, this is the actual cover sheet on your computer, never published. It's circulated to the members of the RNA Tie Club, including James Watson. You can get this online if you go to the archive of Crick's publications. And I highly recommend that you do that. It is just an inspiring, brilliant piece of biological reasoning. So he's trying to answer this question, and he proposes this adapter hypothesis in that manuscript. How is that moving? How is that information moving from nucleic acid to protein? And he begins by describing, in the manuscript that I recommend you look at, he begins by describing the problem. He says, if you look at DNA, and probably he says RNA will be the case, you wouldn't think of them as a direct template, meaning physically interacting directly from the structure of DNA to picking out particular amino acids. As he says here, no one looking at DNA or RNA would think of them as templates for amino acids. Where are the knobbly hydrophobic surfaces to distinguish valine from leucine and isoleucine and so forth? What the DNA structure does show, he says, is a specific pattern of hydrogen bonds and very little else. Now, Crick, to my mind, and he's one of my scientific heroes. I regret never having a chance to meet him. He was a de facto design theorist, and I realize that is a controversial thing to say, but the actual logic of what he's doing is design triangulation. And you can think of design triangulation as putting this principle on the slide into action. When you discover a complex system performing specialized functions, assume that a rational logic and well-matched parts are enabling those functions. Or, more simply, no magic, look for the mechanism, it's there. So he triangulates. DNA, we know, in 1955, carries information, but is chemically nonspecific. Amino acids have specific geometries which require a system to recognize them. We triangulate into C, or to C, within our black box of the cell. What is C? There must be some mediating or adapter molecules that enable information transfer to go from DNA to amino acids. All right, we've got our warrant. Let's go looking for it. And what do we need, by the way? We need 20 specialized molecules, 20 specialized adapters to match 20 amino acids. We would need at least of them. They would need a hydrogen bonding surface because remember, they're going to be interacting with nucleic acid on one side and amino acids on the other. Now, we've got our adapters. We've hypothesized our adapters. What else do we need? Well, something is going to have to attach the amino acids to the adapters in a very, very specific way. Those are going to be enzymes, and we'll need another 20 of them, okay? Because we've got our hydrogen bonding surface, we've got our adapter. At this end, at the amino acid side of the adapter, we need an enzyme to put the right amino acid on the adapter. So here's our adapter. Remember, there's no evidence yet for any of this. This is true deductive biology. He's inferring that it has to be there, starting top down with the system, the no magic principle. It's got to be there. So here's our adapter. There's the binding surface that recognizes nucleic acid. Here is valine. Valine is going to be attached to this end of the adapter specifically because proteins aren't built willy-nilly out of just any old amino acid, and we need an enzyme to do that, and we need 20 of them. 20 adapters, 20 enzymes, 40 parts at least. No evidence yet for any of it. And he realizes this. In that manuscript, he says, we don't have any evidence for this. He doesn't care. To me, this is one of the reasons he's a hero of mine. He just doesn't care. He says, you know what? Cells are not running by magic. I mean, I'm paraphrasing or inferring what's going on. He actually says what you see there on the slide. The usual argument presented against this latter scheme, no such molecules have been found. This objection cannot stand. Bam. He just gives it the back of his hand. I'm not worried about that. Why? Because the system level inference is strong. Cells are not running by magic. We know something has to be doing this. Let's go looking for it. Every time I talk about this example, it takes my breath away because it's real deductive biology, which you usually can't do unless you really understand the system. There are too many possibilities. But Crick, first-rate thinker that he is a de facto design theorist, and he makes that principle work for him. Transfer RNA is the adapter. But notice, it was discovered and characterized 3 years after he predicted its existence in 1955. And when it was discovered and characterized, it had all the features he said needed to be there, such as the anticodon, right? So here's your messenger RNA strand down here. We're just looking at one codon. This is going to bind with that in the ribosome. Up here, 75 angstroms away, that's quite a distance, is alanine at the CCA stem of the tRNA. And this is a true code. This codon via the anticodon means alanine. So this is the chemistry end, the business end of the information transfer that he knew had to be there. And these guys, they are very complicated enzymes where the enzyme, Michael Denton said to me once, what it does is it reaches down under to the tRNA. So the green stem here, or the green ribbon, is the tRNA. This whole enzyme complex reaches down here and says, oh, I see what you are, right? So it's making sure that it has the right anticodon. And then up here, it's attaching the amino acid a long way off. And you need 20 of these guys to do, Crick says, at that time. All right. Why doesn't Watson believe it? Because he didn't. Part 4, and then we'll be done. We'll only run over by a few minutes. Thank you for being so patient. Watson doesn't buy it. Why? Well, because of the implications. Because of the implications of this hypothesis. And this is why you need to call it design triangulation. So in his 2002 autobiography, which describes the period after the double helix, the book The Double Helix is when he describes the period leading up to the 1953 publication in Nature that won the Nobel. And then this book takes the story on from that point. And he says, again, he's brutally honest about himself. He says, I got that manuscript. I didn't believe it. I didn't buy it. Why? Think about this. Why would someone like James Watson, no slouch himself intellectually, very, very bright guy, why would he reject a very plausible and elegant hypothesis like this? Set aside the question of not enough data yet. Just on its merits, why would he say, I don't buy it? Did not like the idea at all, he says. The adapter hypothesis, or excuse me, the adapter mechanism seemed to me too complicated to have ever evolved at the origin of life. What was going on? His biological intuition was chained to a time axis. All right? The time axis necessary in any naturalistic or materialistic story for the origin of life. Looks like this. It's a linear chain of events. You can't start with whole cells. You have to start with their parts. Around their parts, you accrete more complexity and you head towards the living state where A happens, and A has to happen for B to happen, and B has to happen for C to happen. So you have an implicit time axis. And Watson's biological intuition or imagination is chained to that linear sequence of events. In that picture, inferring DNA and proteins and amino acids and all these parts, and then guess what? A mediating chemistry between the two of them, it's just too much complexity all at once. History is just one damn thing after another. If you presuppose that kind of linear progression of events, and that's how cells were built, the kind of inference that Crick makes, where he starts with the whole system and says, what does it need to run? That's going to be highly counterintuitive. And Watson says, I don't buy it, and he takes the wrong path. At that fork in the development of biological theory, he takes the wrong path. And Crick, to his credit, was a much better biologist than he was an atheist. And he takes the other path and predicts the discovery of something that now we know to be the case. Okay. This method of design triangulation makes a demand on the investigator. Namely, you have to start with the system as a whole. You have to start with the upper level, because it's the upper level and its interactions that warrants the inferences to the things that you haven't yet seen. And this is why, and I don't mean any disrespect by saying this, again, he's one of my heroes, but Crick was a better biologist than he was an atheist because he let the biology lead him, not the demand for a materialistic parts first story. And I would say, as I look at his career, the breakthroughs that he had, the insights that he had, the discoveries that he made, always occurred when he let the biology lead him. And where I think he missed the boat, for instance, on his paper in Nature in I think 1980 about selfish DNA, he was letting evolutionary theory and materialism guide him. Again, I mean no disrespect, but he presupposed this, the causal primacy of the organism, the top level first. I think that may be my last slide. It is. Ta-da! We're done, and we're over by 5 minutes. That's not too bad given my track record. Subor, are you there?
Yeah, I'm here. I was just listening intently because some of the things that you've covered today are really interesting from a linguistic perspective, because I remember a few years back watching there's a series called Moving Naturalism Forward. It had all these I'm not sure if you're aware of this 3-day
Sean Carroll, right? It was Sean Carroll was one of the leaders.
That's right.
Dan Dennett was there.
Yeah, Richard Dawkins as well. And one of the things that they were lamenting about was design as a sort of folk language that we're still using. they're trying to come up with ways of not using design because they understand the implications. But what you've done today is actually show it's not just about the fact that we can't get rid of design from a linguistic perspective, but there is a conceptual whole. There's a systems level functional necessity, which is inescapable. So it doesn't really matter if we can linguistically get rid of the word design. You have to start with the system as a whole. And the examples of Harvey and the examples later on about Crick, I think were really, really interesting. I've never heard this stuff before.
Well, I'm glad, because this, the reason I wanted to get this material in at this point in the series about orphan and taxonomically restricted genes is when I come back to them in the next section, I'm going to use design triangulation to illuminate or cast light on, I'm sorry, that's another way of saying illuminate, the role that these play in higher level systems, and how a systems first approach may help us to understand the origin and function of orphans and taxonomically restricted genes in a way that a bottom-up perspective does not. You never want to be in the position as a scientist of blaming the data, right? And I think it's a terrible thing for biologists to have to blame living things for what they are. Living things are what they are, right? They're complex integrated systems. And if you start there, I think nature herself helps you, and she leads you to discoveries. She leads you to knowledge. I apologize for skipping over an example. It had to do with the function of the proteasome, which is the garbage disposal system in cells, and a design triangulation discovery made by a team at the Weizmann Institute. But I'll provide the references for that, and some additional references. I would encourage the listener or the viewer, if they have questions about this, please contact me, care of the Discovery Institute. I have a lot more material on this topic that in the space of an hour and 5 minutes, I wasn't able to include. But let me just say, there's a lot more where this came from.
Yeah, definitely. Could you elaborate a little bit when you were speaking about the direction of causality begins with the system as a whole? Now, that's obviously clear, but the implications of that for the reductive selfish gene perspective, and maybe the interaction between Noble and Dennis Noble and Dawkins comes to mind about systems biology versus reductive approach.
So a good way to think about this is to ask yourself, what is the role of a protein, let's say a DNA binding protein, like a transcription factor? That role is dictated not so much by the chemistry. Yes, the chemistry is important. You have to be able actually to bind a strand of DNA. And when you look at the protein, it has motifs that allow it to do that. But its functional role is way up above its chemical binding capabilities. And the way to come to grips with this sort of as a thought experiment is to realize that the same DNA binding protein can be found in multiple different systems, which as organisms are about as different as animals can be, such as squid, fruit flies, and vertebrates. The DNA sequence and the chemistry for PAX6 Ilus, I forget its name in the squid, it's effectively the same protein, right? So bottom up, you say, well, I've got this module, this DNA binding protein. Now, what is it going to do? Well, you put it in a fruit fly, it will trigger the development of a compound eye, where the optics and everything about the eye is classically not homologous to a camera eye. We have a camera eye, single lens, retinal surface, and so forth. Squid or octopus have the same kind of camera eye, single lens, retinal surface, except their eyes are wired up very differently from ours. They don't have a single optic nerve. Their photoreceptors face towards incoming light, not away from it as ours do. So you'll get two camera eyes, but again, they're not homologous. Those three eyes, compound eye, cephalopod camera eye, vertebrate camera eye, they never shared an eye that was anatomically identical to any of them, right? They all those eyes all evolve separately according to evolution. The same protein, it is the same, and you can do this. You can express a fruit fly, the fruit fly gene in these other systems, or excuse me, you can take the vertebrate gene for PAX6 in the fly and express it sorry, I'm getting a little tangled. Take the gene from the mouse, express it in the fly, and that gene wakes up and it says, oh, I'm in an arthropod. In fact, I'm in Drosophila. Time to trigger a compound eye. The same gene in a mouse, camera eye. So causality is being dictated by the system as a whole, top down, telling its parts what to do. That stands on its head Dawkins' view, which is causality flows from nucleic acid through development into the organism. All of these examples of so-called deep homology. Another would be Michael B. He said to me once, it's a gene distillus. It's another transcription factor, DNA binding a protein. And he said to me once, Paul, it looks like distillus regulates the development of sticky outy things, right? Well, what's a sticky outy thing? Well, it's some anatomical structure that extends out from the main part of the animal. But a sea urchin spine is a sticky outy thing. An insect leg is a sticky outy thing, and so forth. The homology, if anything, is at a very high abstract level of something that goes out, right? But distillus, when you look at the protein itself, distillus is largely the same from one animal system to another. Yeah. So again, and many, many examples like this of lower level module DNA binding protein getting its functional role by asking, I'm personifying these proteins. Where am I today? I'm in an insect. I'm in a echinoderm. I'm in a vertebrate. Okay, my job is going to be different in important ways. So I think evolutionary theory, because of its being chained to a time axis, A, then B, then C, then D, a parts first approach has to ignore a lot of biology that makes perfect sense if you can liberate yourself from that temporal linearity of having to provide that material narrative. And I'll argue this in the remainder of this series. I'll say, intelligent design uniquely of all causal hypotheses lets you start with whole systems. That's what designers do. And I think the history of biology shows the fruitfulness of this method. It's interesting. Harvey made his discoveries, 200 years before Darwin, and they're with us today. The blood circulates.
Yeah, it's interesting because sometimes the arguments made by Darwinists for the truthfulness of Darwinism is essentially the supposed results, the fruits of Darwinism, right? Which, as Jonathan Wells shows, are independent, but still. Well, we could just, if we were to follow that line of reasoning, we could say, well, why is a systems biology approach so much more fruitful than a reductive approach? So doesn't that show that it's more likely to be true? And then obviously, if we go back to a sort of standard definition of design, adaptation of means to an end, well, you have to have an end to begin with, and from that you can actually extrapolate the whole system and how it works. So from an epistemic perspective, there's a treasure trove if you start off with ID, you can actually open up these layers of information. You can actually look at things from a very different angle. However, when we are supposed to be in this epistemic cage and looking at things from a reductive point of view, we're trying to look out at the world, but we have a keyhole approach. We just have a small keyhole and we're trying to work out what things are. It's two-dimensional. I mean, one of the ways, I think you summarized it well going back last year sometime, is we just have more tools in our toolbox.
Amen. Yes. And starting at the systems level doesn't rob you of the facts and realities of the lower level.
Yeah.
What it does is it gives you an additional perspective and a way of integrating the levels. And I think treating living things for what they really are, which are complex integrated systems, where as Dennis Noble says, every level is relevant. There's no privileged level. Dawkins and other DNA first or nucleic acid first thinkers privilege nucleic acid because they're compelled to. And if you talk about being imprisoned in a box, I would never want to have to explain the origin of living things by starting with their bits and pieces. And I think the struggle now in the origin of life, and you'll hear very interesting speakers like John Sutherland recently gave a talk at University of Chicago, where he said, if you go to an origin of life meeting, you'll have camps, people actually sort of standing in different corners of the room, metabolism first, nucleic acid first, and tenaciously defending their particular take on things, what characterizes that logically? What characterizes that is privileging some bit, privileging some piece. It had to be metabolism. No, it had to be inheritance. It had to be nucleic acid, right? And the design theorists can say, why not say it was the cell? Why not start with that? Well, that's design, right? That's off the table.
Yeah.
And I think the challenge for design, the design community, is to get their ideas to a point. And I've seen this at 2 meetings this summer. I'm just thrilled by the young people who are working in this area, who are taking design as their motivating heuristic and saying, you know what, I can illuminate this system or that system in light of design, and I can take what I found to peer-reviewed journals.
Yeah.
They have to be very careful about it, because you can engender a lot of opposition if you say intelligent design right there in your abstract. But in fact, that's what they're doing.
Yeah, absolutely. You've summarized it really well. I'm sure everybody watching is going to be looking forward to the next episode that we actually have, which is probably going to be the final one.
Yeah.
So thank you so much, Paul, for your time. It was a lovely session. For everybody watching, Assalamu alaikum wa rahmatullahi wa barakatuh.